Hello, my name is Ege Kaan Arıcı, and I've done my research on investigating the relationship of PFOA resistance and the expression of two dehalogenation genes in P. petita and P. aeruginosa. Perfluoroalkyl substances, otherwise known as PFAS, are a family of pollutants characterized by their chemical structure made up of long carbon chains bonded to fluorine atoms to form one of the strongest chemical bonds in nature. Due to the CNF bond, these substances are exceptionally resistant to degradation under natural conditions and are highly toxic to many organisms due to their abilities to accumulate in bodies of water and soil. Previous studies have shown that the genus Pseudomonas, which are made up of gram-negative non-motile bacilli that have the ability to grow over wide ranges of temperature and various growing conditions, contain some organisms that have the tools to not only grow resistant to PFAS, but also degrade them under certain conditions. In this study, the relationship between the expression of two dehalogenation genes, LDEX and DH1, were investigated using qPCR and supporting tests such as optical density at 600 nanometer readings and NPN assays. The OD 600 and NPN data suggested an increase in the resistance of PFOA over time and with increasing doses. However, the qPCR results showed a significant downregulation in the LDEX and the EH1 genes for P. aeruginosa strain and no significance in P. petita strains. The methods include both bacteria being initially grown on nutrient agar and then for 24 hours under 37 degrees Celsius and then being transferred into test tubes containing 10, 50, 1 and 100 milligrams of liter PFOA in those test tubes. These cultures were incubated in a Schenker incubator for 37 degrees for 4 days while the optical densities were read every 24 hours. After 4 days, the cultures were tested using NPN assays to see viability, and the bacteria were replated for growth in a 37 degrees incubator. This procedure was repeated twice more to result in a total of 3 generations. Finally, at the end of the week 3, the 100 milligrams per liter treated culture were analyzed by qPCR. The optical density readings at 600 nanometers shows an increase in bacterial concentration over the 4-day treatment period, as well as the 3 generations for P. petita and P. aeruginosa. It also shows a larger increase in bacterial concentration for P. petita compared to P. aeruginosa, as well as higher doses of PFOA treatment compared to the control cultures. These results indicate an increase in PFOA resistance by both strains, especially for P. petita. The NPN results show an increase in viability for both P. petita and P. aeruginosa throughout 3 generations and with increasing dosages of PFOA compared to the control groups. Throughout the 3 generations of both P. aeruginosa and P. petita, the amount of bacteria has been in 10 to the power of 9 for the control groups, while the treatment of 1, 10, 50, and 100 milligrams per liter has shown 10 to the power of 10 in the first generation and has increased to 10 to the power of 11 in the second and third generations. The qPCR results show that there was a significant decrease in the expression of LDEX in the H1 genes for P. aeruginosa, but no significant change for LDEX and the H1 for P. petita compared to the housekeeping gene 16S rRNA. This result indicates that neither LDEX and DH1 had a part in PFOA resistance ability of P. petita or P. aeruginosa strains. Overall, the conclusions for this experiment suggest that the results were conflicting, and then bacterial resistance against PFOA and PFOA by P. petita and P. aeruginosa were not related to the dehalogenation genes LDEX and DH1. The OD600 and NPN assay data indicated resistance over time to PFOA, increasing at high doses. However, the qPCR data indicated that the DH1 dehydrogenation gene and LDEX dehydrogenation genes for P. aeruginosa and P. petita were not related in the mechanism of degradation nor resistance. For the future directions of the experiment, if this was to redone, to find a way to detect the byproduct of degradation for PFOA over time, such as mass spectrophotometry, and test other genes that might be related to the degradation and resistance mechanism of bacteria. Thank you for listening.